what all of you have waited for, the movement into our first physiological topic. Oh my gosh, you guys, I know you're so excited about this. So, before we get into our first, excuse me, discussion of physiology, we're going to move away now from kind of the gross anatomy of the heart and start getting down into the microscopic anatomy of the heart. So, when we look at cardiac tissue under a microscope, if you remember back to AMP1, and you went back to one of your favorite chapters, histology, okay, you may have remembered that when we looked at our muscle, whether it be skeletal muscle, cardiac muscle, or smooth muscle, two of those tissues were striated, right? So our skeletal muscle was striated, and those striations came from the appearance or the presence of actin-myosin. Our cardiac tissue is also striated, which then tells us that we have a structural organization very similar to our skeletal. However, one thing that's different, or a few things that are different between our cardiac muscle and our skeletal muscle, is our cardiac muscle has branching. So what you can see here is these cardiac muscles will branch down into other muscles. So they have a branching appearance to them. They tend to have, or they have, not they tend, but they have these structures known as intercalated discs. I'll talk about those a little bit more here extensively on the next slide. They typically will have 1 to 2 nuclei. Okay. They have a T-tubule, just like skeletal muscle did, right? So here's our T-tubule. Remember, our T-tubule is what allows for that electrical signal to come down inside the cell. However, they're a little less numerous than they are in skeletal muscle. Their sarcoplasmic reticulum. They contain a sarcoplasmic reticulum. Again, not as quite as complicated as we see in skeletal muscle, but that sarcoplasmic reticulum is still going to contain calcium, just like in our skeletal muscle. Okay, and they have a massive number of mitochondria, which tells us about 25 to 35% of the cell is actually made up of mitochondria. So again, they have a very similar structure. We can see the I band and the A bands. We see the Z disc. We see the actin and myosin appearance. Okay, this is all the same. Okay, so again, one major difference or two major differences: they branch and they have the presence of these intercalated discs. Now, these intercalated discs are basically sites of junction between cells. So where you have an intercalated disc, for example, right here, you have one cell ending and another cell starting, and they're linked together at this site of an intercalated disc. Now, these intercalated discs have two major cell type junctions that help the heart function as what we call a functional synctum, or as a single unit. So we have the presence of desmosomes. And if you remember from ANP1, desmosomes are really good at preventing shear stress. So as that heart contracts, right, it's a very, very dynamic process. What happens when it contracts, those desmosomes prevent that heart from ripping apart as it's contracting. In addition to that, those intercalated discs also have the presence of our gap junctions. And what our gap junctions allow is it basically allows ions to move freely from one cell to another very rapidly. Okay? So what it essentially does is it couples the cells. It basically creates a uniformity in our cells and their ability to contract. So unlike skeletal muscle, where we have a recruitment based on size principle, based on demand, the heart, we want the heart to all beat at once and then relax at once, and beat at once and relax at once. So we really need it to act and behave like a single coordinated unit, unlike our skeletal muscle, right? Where if we want to go and gently rub the head of a child, we don't want to recruit our large muscles as well as recruit a huge number of those motor units. Okay? The heart's different. It all wants to coordinate as one single unit. So let's review skeletal muscle. And you're like, well, we're not talking about skeletal muscle, but I want to bring it up. Okay. So when we talked about skeletal muscle, right, we drew this awesome, we're not going to draw it super specific. We drew this awesome action potential. coming down our axon terminal, right? So we have this action potential that comes down, right? And it lands on, I'm just going to kind of draw a tube here. Okay. I don't have the synaptic cleft here, but you guys get the gist of it, right? Here, this is our skeletal muscle cell. Okay. Our action potential comes down, right? So this is essentially our electrical signal. Okay, it comes down. We have an end plate potential that causes our action potential to propagate down our sarcolemma, right? It goes down our T-tubule, right? Releases, gets our sarcoplasmic reticulum, causes that to release calcium, and this then goes and targets our actin and myosin, and we get a cross-bridge cycle. Oh my gosh, so exciting, right? Okay, hopefully you guys kind of remember those events. Okay, I'm actually going to erase this because it's actually not what I really wanted to draw here, but I just wanted to remind you guys of how that occurred. Okay, so if we look at the action potential tracing of those events we just talked about, okay, we have our axis that looks like this. Okay, we have our cell at a resting membrane potential. Just kidding, minus 90 in a muscle. Okay, and what's going to happen is we get a stimulus here, right, that causes a depolarization. Okay, at the peak, we get a repolarization, and we go back to baseline. Okay, now this depolarization event in the case of a skeletal muscle, again, remember we're referring to a skeletal muscle, results or is caused by an influx of sodium into the cell. Okay, so if we're looking at the inside of a cell here, right, in this case sodium is going into the cell, okay, and causing this to become positive, right? Our repolarization, so at our peak, sodium closes. Hopefully this is coming back to you guys. Potassium opens, right? And our repolarization is due to a potassium exit, or efflux from the cell. So in our repolarization, potassium is leaving, causing the cell to go back to its negative state. Okay? Now, I want you to keep that in mind as we move forward. Sodium causes depolarization, potassium causes repolarization. Okay? So just shelf that for a little while. Okay? Okay. So when we look at the similarities between skeletal muscle and cardiac muscle, so now we're going back to cardiac muscle, but because we know skeletal muscle, hopefully, we're going to use that as a comparison. So our similarities, okay? Muscle contraction is preceded by a depolarization action potential. Now, in skeletal muscle, it's a nerve. In cardiac, it is not. So don't think about that. No, that's why I didn't, on that picture originally that I drew here, I didn't label that little axon terminal, because I just wanted you to think of it as an action potential, as an electrical signal. Okay? So, skeletal muscle needs an electrical signal to activate. Cardiac muscle needs an electrical signal to activate. Okay? That depolarization wave, okay, travels down the T-tubule, causes our sarcoplasmic reticulum to release calcium. Cool, right? We already know that. Same in cardiac. The release of that calcium binds troponin, right? There's our lock, right? Binds troponin, moves tropomyosin, actin and myosin combine. We get our cross-bridge cycle. Oh my God. Sliding filament model, contraction. Awesome. Okay? All the same. If you aren't sure what I'm talking about, make sure you go back to chapter, I believe it's 8, or the skeletal chapter, and there's a really nice picture of that process. Okay? That's all the same. Skeletal and cardiac, identical. Here are the differences. Because of course there's going to be differences, otherwise we'd call a cardiac muscle skeletal muscle. Okay? So the first big difference is all cardiomyocytes. Cardiomyocytes and cardiac cells are the same thing. Okay? Are going to contract as a unit or none do. So it's an almost like an all or none. The entire heart is like an all or none. Okay. Cardiac muscle has an absolute refractory period. Let me go back really quick to our picture. Remember, when we talk about the absolute refractory period, okay, again, hopefully you guys remember what an absolute refractory period is, okay. An absolute refractory period is the period in which no matter how strong a stimuli, you can't generate another action potential. So this is our absolute refractory period. Okay? So in skeletal muscle, it's really short. In cardiac muscle, it's actually fairly large. And I have a picture here I'm going to show you in a minute what I mean by that. The third difference is the heart is actually comprised of two cardiomyocyte populations. We have a contractile population, which is the bulk of the heart. I call this population, let me go back to my black pen. Okay. I call this population my 99%, the contractile population. Okay. We also have a non-contractile population, okay, that is called the pacemaker cells, and I call this my 1%. Nobody else calls these 99% and 1%. I just think of it this way so I can explain it a little bit easier later when I'm talking about how these cells right here, these pacemaker cells, depolarize, okay? So, contractile population, non-contractile population. Contractile makes up most of the heart. Non-contractile makes up a very small portion of the heart, okay? Now, the unique and crazy thing about this contractile population, or this non-contractile, the pacemaker cell population, is they spontaneously depolarize. Oh my gosh. So where a skeletal muscle and our contractile muscles require this stimuli, our pacemaker cells don't. They do not require any nervous system stimulation. Insane, right? Like, they can depolarize the entire heart without, you can take a heart out of a person and it will beat in your hand. And that is because of this pacemaker population. Again, we'll talk about that here in a few minutes. Actually, we're going to talk about that in the next video. I don't want to get too far away from this contractile population. Okay? So, let's go back to the 99% before we jump forward and talk about the 1%. Let's talk about the 99%. Okay? So, continuing with our differences, depolarization wave allows slow calcium channels in the sarcolemma, which then causes the sarcoplasmic reticulum to release calcium as well. Then we get this calcium surge that prolongs our depolarization phase. So let's look and see how this works. Okay. So I like to draw this out. Let's go back to our drawing here. You guys okay with that? Okay. So, and then usually in a classroom I would draw these next to each other. So I'm going to draw this one here, and we'll go back down to the slide here in a minute. Okay. Get rid of that. Okay. So, again, this is our skeletal muscle over here. So we're going to rewrite this. This is going to be skeletal, if my pen would work. There we go. Okay, skeletal muscle. Ah, sorry, that was really loud. Okay, over here we're going to do cardiac. And I'm going to put in parentheses my 99% because I just want to. Okay, so we're going to draw the same profile, or the same kind of y and x axis. We're going to draw our cell down here. Okay, and so we talked about the fact that we still need a stimuli. Okay, so we're going to get a stimuli. There's our stimuli, electrical stimuli. Okay, we are still going to get a depolarization. Okay, however, up here we're actually going to get a plateau. I don't think that's, I think it's plateau with EU. I don't even know. and then this repolarizes. Okay, so now we can start to see the difference between these two. Okay, so in our depolarization of our cardiac cell, okay, this is still caused by an influx of sodium. Whew. Same, right? So sodium in, right, causes us to become positive. Whew, that's not crazy. Okay. However, sodium closes here. And when I say sodium closes and sodium opens, the channel. Okay. And what happens is what opens here is our calcium influx. Okay. So this is new. So what happens now is we also get an influx of calcium. This here causes that plateau. Okay. And then we have our repolarization. This is due to potassium exit, just like it is in skeletal. Okay. Our potassium exits, and our cell, inside of our cell, becomes negative. Okay. Now, if you look at that refractory period, okay, look what we just did to that refractory period. We took it, right? Whoops, oh my gosh, my lines are horrible. Okay. And it was originally this wide, right? And now, look how wide we've made that refractory period. Is that not the coolest thing in the world, right? And the reason we've done that is, the reason you need this plateau is you need to give the heart time to complete its entire contraction before you reset the system. So that plateau allows for all the blood to be ejected from the blood, or from the heart, before the next action potential starts. Is that not so cool? Okay. Okay. So we can see how those are similar, okay, but yet different. And we can see that here. Okay, so this shows it just again in the same schematic. I just like to show it next to the skeletal muscle because hopefully most of you or some of you feel like you kind of remember the skeletal muscle, so you can see it next to each other to really see those differences. Okay, the only thing that's really added in this graph is that tension, the contraction, right? And you can see that, right? This contraction event is fairly stark and is needed in order for us to be able to initiate the next depolarization event. Okay. So again, what I didn't put on the other one is, in order for us to repolarize we have to inactivate the calcium channels and open our potassium channels. Okay. That calcium is then pumped back to the sarcoplasmic reticulum as well as extracellularly. Okay. Okay. Are we doing okay so far? Okay. I just realized I clumped the pacemaker population into this lecture as well. Okay, so what we're going to do now is we're going to talk about that 1%. Okay, so let me remind you what I said a few minutes ago. The heart can depolarize in the absence of the nervous system. The heart can contract in the absence of the nervous system. Okay, so again, the coordination of the heartbeat comes from the fact that we have those gap junctions that allow those ions to move freely in between cells, okay? But also because of our pacemaker population. Those two things allow us to have that functional symptom, that coordinated heartbeat, okay? So this non-contractile population, again, oops, let me go back to a black. Is this 1%. It's still a cardiomyocyte. It's still a cardiac cell. It's just slightly different in that it doesn't have a contractile ability. Okay. So these, what we call autoarrhythmic cells, these pacemaker cells, they have a really unstable resting membrane potential, something called a pacemaker potential. And this is due to the opening of slow sodium channels. So what happens is they have the ability to continuously depolarize. Okay. Now, I'm just going to jump forward because I'm going to explain that slide here. Okay. So again, this is our 1%. Okay? So here is our action potential profile. Okay? And what you can see automatically is we hit this low spot here. And then all of a sudden we get this slow trickle up. And what happens is when this cell meets threshold, it depolarizes. So this is our depolarization. And this is our repolarization. Okay? So what's happening is the following. This leakage here is due to a sodium influx. Okay? Now, it's not a rapid influx like we see with the 99% or the contractile population or in skeletal muscle. Okay? This is due to kind of like, you can kind of think of it as a leakage channel in some aspects. Okay? But this movement in a positive direction is due to sodium. Okay? Well, that means that this depolarization event can't be due to sodium. Okay? Instead, this depolarization is due to a calcium influx. Okay? So we hit threshold, this calcium channel opens, a whole bunch of calcium runs, or basically flows into the inside of the cell. We get up here, calcium channels close, right? Potassium opens, and we repolarize. So this is potassium exiting. Okay, we get down here, potassium closes, and here our leakage starts again. So this region from here to here, the sodium leakage, is what we call the pacemaker potential. Okay, and that is because that is what's creating essentially, ultimately, the action potential. Okay, now we talked about the fact that 99%, the contractile population, requires an electrical signal, right? We said that. This is that electrical signal. So you have a population that doesn't contract, but that can depolarize on its own, okay? Depolarization, right? That can send a wave. So if this pen is the 1%, right, it's going to depolarize on its own. It's going to send that electrical signal across my hand, which is my cardiomyocytes, and cause these ultimately to contract. The heart is so fascinating. Okay? So, the location of this 1% varies throughout the heart. So, these cardiac pacemaker cells exist in these different nodes throughout the heart. So we have the sinoatrial node, we have the atrioventricular node, we have the atrioventricular bundle, the right and left bundle branches, as well as the subendocardial conducting network, which I will always call Purkinje fibers. I will never call it the subendocardial conducting network, because when I learned it, they were called Purkinje fibers. And I still can't wrap my head around this here, as you can tell, by me being tongue-tied by it. Okay? If you give me subendocardial conducting network, I'll still give that to you. But I will always call them Purkinje fibers. Okay? So, here's our heart. Okay? And here is our sinoatrial node. So our SA node is located in the upper right atrial wall. Okay, it depolarizes faster than the rest of the heart. So all of these other yellow regions are the other part where we have that non-contractile population. And so because it depolarizes faster than the other ones, it kind of sets the tone or the rhythm for the heart. So this node will depolarize about 75 times per minute. Okay, as that depolarizes, it sends that impulse across the atrium and across the atrium, both the right and the left atrium. Okay. Now, it also is going to send a signal down to our next node, which is the atrioventricular node. Our atrioventricular node is located on the inferior aspect of the interatrial septum. Okay. It delays the impulse slightly, and the reason it delays is the delay really allows for the atrium to contract, okay, before it initiates the depolarization event in the ventricles. Now, if you lose your sinoatrial node up here, okay, if that stops working, your atrioventricular node can take over. However, it beats or it depolarizes at an inherently lower rate. So it actually only depolarizes about 50 times per minute in the absence of the sinoatrial node. Now, you'll never see that if this SA node is working properly. You only see that if the SA node starts to fail or fails. Okay. So from our AV node, so again, we have our SA node here. We go down to our AV node. Okay. And that AV node then depolarizes or sends that electrical signal down to what we call the atrioventricular bundle, also known as the bundle of His. This is the only electrical connection, this right here, only electrical connection between the atrium and the ventricles. And that is because there are no gap junctions between the atrium and the ventricles. So this wave of electricity that's going through the atrium up here from the SA node can never reach the ventricle, okay, because there's no gap junctions there. Okay, so the only way for the ventricles to be able to get an electrical signal and ultimately contract is through this mechanism here. It's from our AV node to our bundle of His, or atrioventricular bundle. Okay. From there, our atrioventricular bundle, or our bundle of His, branches out into the right and to the left bundle branches. Okay. This basically diverts what ultimately is going to be a signal to the left side of the heart or the right side of the heart. Okay, so this carries the impulse to the apex of the heart. And then we enter into the Purkinje fibers, okay, which ultimately comes down and basically turns and goes up into the ventricle wall, okay, and allows for contraction of the ventricles, or the depolarization of that contractile population in our ventricles from the apex upwards. So this depolarization event will cause a depolarization in this direction, which will then cause a contraction to follow in this direction. Now, I can't iterate enough. Like you guys remember, the whole purpose of depolarization is contraction. Okay? So you have to have depolarization to precede contraction. If you don't depolarize, you can't contract, right? You can't shorten that muscle. Okay? So what we ultimately have is depolarization of our non-contractiles causes depolarization of our contractiles, which causes a contraction. Okay? And we can see that here. Okay? So here is our pacemaker potential. Right? So sodium, calcium, potassium. I almost put P. A P for potassium. It's supposed to be K. Okay? So here's our SA node. Okay? And what we can see is this depolarizes. Okay? This depolarization causes our atrial, right? This is a stimuli. Our SA node triggers that stimuli, that spread across our atrium. Okay? Our atrium depolarize, which means following that depolarization, don't forget, let me use a different color. Don't forget, if I wanted to superimpose on this, right, I would have a line that looks like kind of like this. That would be contraction of the atrium. Right? Okay? And remember, we have a slight delay, right? This AV node and this cardiac muscle don't depolarize at the same time, even though they're getting the same signal at the same time. And again, because remember, there's that slight delay so that the atrium can actually finish contraction. Okay, our AV node depolarizes. There's our signal to the ventricle, right? That goes down the bundle of His to the right and left bundle branches up the Purkinje fibers, and that sends that depolarization signal out to our 99%. So, right, so this is the 99% in atrium. This is the 1% in atrium. Right, this is our 1%. This is still kind of in the atrium, but the only way to get to the ventricles, and this is our 99% in our ventricles. Right, so this atrioventricular node, right, what we don't show here is the bundle of His and the left and right bundle branches and the Purkinje fibers, but ultimately this triggers this big, massive contraction of the ventricles. This all happens in a single heartbeat every single time our heart beats. It's insane. Okay. So some homeostatic imbalances that we see in patients in terms of their intrinsic conduction system, that's what this, so when we talk about, I don't think I mentioned that yet, this AV node system right here, okay, this group of structures and process, this is known as the intrinsic conduction system. So this is intrinsic conduction. Okay, so you may see that pop up, or I may say it not thinking about it or explaining it to you. Okay, so if there's a defect in one of these systems, okay, this can lead to an arrhythmia. An arrhythmia is just an irregular heart rhythm, okay, and typically results in an uncoordinated contraction of the atrium and the ventricle. So you may get 2 contractions of the atrium to 1 contraction of the ventricle. You may get 1 contraction of the atrium to 2 contractions of the ventricle. So you get these uncoordinated kind of events that can occur. Now, there's also something called a fibrillation, okay? And these are rapid irregular contractions. They are completely useless for pumping. If it occurs in the ventricle, okay, if you have what's called VFib, ventricular fibrillation, this can lead to basically issues with circulation seizing and ultimate death, okay? AFib, on the other hand, which is atrial fibrillation, most people can live with just fine. You can take some meds for it, maybe, maybe not, okay? But when someone goes into VFib, typically what you'll do is you'll use defibrillation to treat them. And defibrillators, you can find those in like grocery stores. We have a defibrillator in the hallway out by the 4-way on campus. It's essentially when you electrically shock the heart back into a proper rhythm. Now, you can have a defective SA node, right? That first node, that first firing node. Okay, and this can cause something known as ectopic focus, which is basically an abnormal pacemaker. The AV node takes over, okay, which is fine, and sets a new rhythm known as the junctional rhythm. So in patients where you have ectopic focus, okay, their heart rate tends to be slower because the AV node, remember, only fires about 50 times per minute versus the SA node, which is about 75 times. But it's okay, because we can still, the body's still getting enough oxygen and nutrients even at 50 times per minute, okay? You can also get something called extrasystole. This is a premature contraction. So sometimes an ectopic focus sets a high rate, and this can be excessive. For example, sometimes people will drink too much caffeine or have too much nicotine and they feel like their heart is racing, right? That's because of this event, okay? Now, in order for, again, that signal to reach the ventricles, that impulse has to pass through the AV node. Remember, so it goes SA node, AV node, bundle of His. Well, what happens if you don't have an AV node, or you have a defective AV node, or your AV node fails? Can we get a signal to the ventricles? No. Right? So this is known as a heart block, and there's different degrees of heart block. So sometimes it's not a complete failure, which is what you ideally want. I mean, if you're going to have an SA node or an AV node go bad, you want at least to have some signal going through so that your heart still contracts. Okay? So if you have a total heart block, that means your ventricles will never. So So the bundle of His and the bundle of His actually has an intrinsic depolarization. However, it is not fast enough to sustain life. So it's not like if you get an AV block or have an immediate damage to an AV node, it's not like the heart just stops. Your heart rate just goes so low that you can't sustain life. Okay. So what they'll do with these patients is they'll implant an artificial pacemaker. And this artificial pacemaker will essentially serve as that trigger to the ventricles. Okay. So in terms of, so I've just told you that the heart doesn't need nervous system to depolarize. Okay. However, in order to increase, so we said the AV node or the SA node sets the pace at 75 times per minute. Okay. However, there are times you need that heart rate to go much faster, right? You're out in the woods and a damn bear is chasing you, right? You're not going to get very far if your heart's still just like, I'm just cool, I'm just going to be 175 times per minute, right? Okay. So we have ways to adjust that throttle, okay? And it does it through the autonomic nervous system. Okay, so, oh my gosh, you guys go back to chapter 11. No, autonomic was chapter 14. Go back to chapter 14. We're talking about autonomic nervous system. So this is our parasympathetic and our sympathetic nervous system. Okay, so we have these cardiac centers in our medulla oblongata, and these cardiac centers are signal, not are controlled by, but it's signal through the autonomic nervous system. Okay, so if we activate the sympathetic nervous system, fight or flight, okay, we increase heart rate, we increase force of contraction of the heart muscle. That makes sense. Okay, and that is because we've activated the cardioacceleratory center in our medulla oblongata. Okay, and this is going to affect, for example, our SAV node, our AV nodes, our heart muscle, and our coronary arteries. So that cardioacceleratory center via the sympathetic nervous system has a significant effect. Okay, now if we need our heart rate to decrease, like there are ways to decrease that heart rate. Okay, so you'll hear about people who meditate and are able to decrease their heart rate. And that is because they can kind of harness and activate the parasympathetic nervous system, this arm of it, and this will decrease our rate. So in that case, we actually activate the cardioinhibitory center. Okay, and that parasympathetic nervous system inhibits our SA node and our AV node via the vagus nerve, just kind of slowing that intrinsic contraction down. Okay, so again, a lot of physiology. Make sure you guys go back and look over that, okay? And email me with any questions. I'll be right back with the next video.